firstly, hello everyone. I'm Nate, and a bit of a disclaimer. You know, whilst I work at Google, I'm not part of the team at all, and only have the same access the public does to anything related to AlphaGenome. So any opinions are purely my own. So I'll be running the session along with David. David, do you want to introduce yourself?
Yeah, hi everyone. Thanks for joining. I'm David Kaner. I'm a senior research fellow at UQ, University of Queensland. And yeah, one of the co-organizers working with Nathaniel on this, and bit of a background in computational biology and AI, but you know, this stuff is also pretty new and out there for me at times. So this will be an interesting discussion, I hope, today.
Fantastic. So yeah, to keep the sort of discussion going, please post any comments you have in the chat, and we'll sort of take a moment to capture any of those queries. Otherwise, we'll make sort of frequent stops to hear from people and your opinions. So yeah, like I'm by no means a geneticist or even a biologist, but AlphaGenome seemed pretty exciting to me. So I wanted to sort of understand the implications about what it could do, and you know, bear with me because I might introduce the context that I've discovered. Maybe that'll help a few newer people on the call. And I'll just post some links in the chat. Here we go. Those ones. Oh, wait. Okay, there's half of them. A couple more, maybe. Good stuff. Yeah. So if you need to get a copy of the paper and so on, there's quite a few links there, but feel free to go to them as at your leisure. Especially this notebook LM has been super helpful for me. You know, I just ask it questions when I don't understand something. I've thrown all the documents in there, so you can ask questions as we go along. And it has a link to the paper in there as well. And yeah, so my context coming at this from as a non-geneticist. So the genome, it's the full set of DNA that shapes sort of how an organism and its traits, growth and function and so on. And DNA is made up of the 4 nucleotides, A, C, G, T, the letters, and genes are the discrete sequence of these letters, and they can be from sort of hundreds to millions of base pairs long. And only about 2% of our DNA codes for proteins, and the other sort of 98%, that's the non-coding regions. And that does things like regulate gene activity, chromosome structure, and sort of other things as well. And both are really essential. And AlphaGenome, you know, focuses on that 98% of non-coding regions. So genes have start and end points, and their activity depends on processes like splicing, RNA production, and DNA modifications. And small genetic mutations, they can change how genes work, affecting things like health and disease risk. So AlphaGenome, this helps to decode all this regulatory system by predicting how these little genetic variations might impact the gene's various biological functions. So please, you know, feel free to correct me or anything if my understanding is not quite there. But yeah, the implication is that AlphaGenome can help us with things like revealing disease mechanisms, aiding synthetic DNA design, or speeding up genomic research. And AlphaGenome, it's somewhat complementary to another tool called AlphaSense, which was released a few years ago. And that focused more on the variants within protein coding regions. So as I said, AlphaGenome specifically sort of targets and wants to interpret the implications of variants in the 98% of the genome that are non-coding. So that was my kind of context. And the paper is purely about the tool itself. Like there's no new discoveries here. It's fully a methods paper. This is my takeaway from it. So this is the preprint. I believe they're working towards formal publication, and it's probably under review, I assume. And some things that that I pointed out. Oh, sorry, that there were kind of 4 questions that I wanted to answer today. If people have anything else, please put your questions in the chat. So what is AlphaGenome? How does it work? What can it do? And also what can't it do? And I thought we could talk for like about half an hour or so, and we'll give us about half an hour to do a Python demo and actually demo the tool itself. Good stuff. How are we doing? Anything yet? Good. Oh, we've got like 77 people. Love it. So
hey, before we go on, Nathaniel, do we want to take questions along the way? So I noticed someone just raised their hand.
Oh yeah.
So, and I'm not sure in the Google Meet system how we actually see. That was Tom Hill. That was an error. But nevertheless, it was good timing, Tom. Because do we want to kind of make it a sort of, if people want to sort of raise their hand at any point, or
absolutely, I can keep talking all day, but yeah, definitely not the expert. So I'll keep riffing it anyway, but please interrupt as we go. Right. So yeah, the things that I kind of highlighted, AlphaGenome takes in 1 megabase, a million base pairs of DNA sequence, and predicts sort of thousands of functional genomic tracks up to a single base pair resolution. So that's a huge sort of step forward in what has been here before. And it's got this huge range of modalities. So gene expression, transcription, initiation, chromatin accessibility, histone modifications, transcriptions for factor binding, chromatin contact maps, splice site usage, and splice junction coordinates and strength. I don't know what a lot of those are, but hopefully they're important to everyone on the call. And it's been just trained on human and mouse genomes, so keep that in mind. When I was talking to David earlier, I was like, oh, you're cool, so you can just retrain this on plant data, and he said no, it's not that easy. I believe the weights of the model will be released when the formal publication comes out. And so AlphaGenome has matched or exceeded basically the models that are out there already. So it's done a good job. Now, what else is interesting?
So we have a question, Nathaniel. Simon it says, a newbie question: AlphaFold was massive in terms of impact. Is AlphaGenome an advance on that same scale? Which I think is a tricky one to. I personally wouldn't view it on the same scale as AlphaFold in terms of impact, because there are other sort of DNA models. Certainly there are. I work in plants, and there are certainly quite a few in plants that do a similar thing. I think where AlphaGenome is happening is it's increased the accuracy. It's pushed the accuracy of these kinds of models to a new level. But compared to the level that AlphaFold extended protein folding accuracy, it's not as big a jump as that.
Yeah, so figure 1 in the paper shows sort of a comparison between the state of the art models. And so one thing AlphaGenome does push forward a lot. I mean, it beats a lot of these benchmarks, some by a considerable margin, most of them just by a little bit, but it brings them all together. I think that's where the power comes in. So yeah, these are all different tools that perform, different sort of specific or target different modalities, and yes, some of them it matches, and some it far exceeds, but it does all of them. So that's the really cool thing about it. So both in the track predictions and the variant effect predictions. Okay, yeah. So what else is interesting about what AlphaGenome is? Which we've sort of has come up, that it's this thing called a sequence to function model. I hadn't heard that term before, but it's exactly what it sounds like. It takes a DNA sequence, and at the model, so the deep learning model outputs a series of like functions and parameters and different sort of data types, which I find really interesting. You know, like most models are kind of like text to speech or text to text, but this is a DNA sequence to various different modalities, which is really interesting. And I think that comes up in the training that they've done on this model. So let's go have a look at some of that. Again, in figure 1, the input for, oh sorry, this is what the model predictions look like. So you give it a DNA sequence up to a million base pairs, 1 megabase pair, and it goes through what's called like it's inspired by a UNet, and it's called a UNet because when you write it out, the resolution of the model gets sort of it starts very large and then it gets very small and it gets very large again, so it looks like a U. And basically it steps through the model, goes through a convolutional neural network, and it learns different traits and different it finds sort of different connections in the data as it goes through the different steps down through the model, and then as it comes back up through, and then it has this variety of different output types here. So there's all these different output types, and the training to go into this was kind of a large DNA sequences, but then they did a whole bunch of perturbations to the DNA sequences. So they do like random shifts, or they'd bring in sort of different known variants. They'd bring in I don't know, like they did heaps and heaps of different types of training, and they used something called a distillation process, or a teacher-student method, where they taught from the raw data. They trained a whole bunch of models, but then they used those models which are the teachers, to then train a student model. And I think that must get rid of like a lot of maybe the noise or something in the model, because these have been shown to perform much better than just doing a sort of a straight train model, these teacher-student models. Any questions? How are we doing? So this this figure is really well explained, I think, in, I might open this, can I open it in new window? Yeah, in the supplementary material. So way down the bottom, or hopefully everyone's read all 103 pages.
Nate, do you want to take questions as they come up?
Yeah, for sure.
So Irina has got a question.
A quick question. I was wondering, so what's the main improvement in the architecture of the model over the Informer and Borzoi? Are these the student-teacher models or the decoders?
I'm not sure how both Informer and Borzoi work, but so figure 1 in this, is it figure 1 in the supplementary sort of describes the model in more details, but yeah, I don't know how Borzoi and Informer work, but I know that they don't have as many output modalities as well.
Yeah, that makes sense. Thank you.
Yeah, so if you want to unpack the sort of the UNet in even more detail, it's sort of described in further detail here, and then also in the method section, you get the pseudocode that describes it, and they may release the code eventually. Yes. Okay. So that's more or less the model architecture. Is there anything else interesting in that? So yeah, whilst it's 103 pages, the first 17 pages I think are really striking, and then there's a whole bunch of methods, and then there's a whole bunch of supplementary figures, so it's not too overwhelming. Yeah, and then they go through and describe the various training methods. So there's nothing really like groundbreaking. Nothing stood out for me in how they sort of did anything, but it was just they did so many little sort of techniques and training tasks to bring everything together. So I really think that's the power of the model. And it's quite lightweight for inference. So at the moment, the only way to access the model is via the API, which we'll talk about. But yeah, it takes like a second of computational time, which works out to be relatively cheap in terms of costs. But for actual training the model, I think they use a distributed tensor processing unit, TPU, sort of like a GPU, which is far more rigorous. So I think there were sort of dozens or hundreds of GPUs that they use, or TPUs. Okay, so what else is interesting? Yeah, so it's state of the art and all these different variant effect prediction approaches. Is that figure 2? Yeah, so figure 1 sort of gave an overview of how the model works and its benchmarks. And then figure 2 here, it just explains or shows how the model performs in its own right. So we have just an example track prediction, and then it has the observed versus the predicted. And so I don't fully understand what all these different metrics are, but this is the observed and this is the predicted. They're looking fairly similar to me, and then they unpack these in different parts and look at it in finer resolution, just to prove that like the model is performing, is predicting at very close to the observations.
Nate, we've got a question from Pratap.
Oh, yep.
He's raised his hand. Pratap, if you'd like to ask your question. Not sure if, how do you, I can see the raised hand queue. How do I inform Pratap that?
Yeah, sorry, I pressed it wrongly.
Sorry?
Oh, you pressed it wrongly. That's all right.
Yeah.
Oh, okay. No worries.
That's all right. Cool. So yeah, so that's figure 2. Yeah, just proving that the model works compared to observations, and we keep going through. What's interesting now? So then now they sort of start comparing the model to a couple of the other tools that were or are state of the art, specifically the variant effect predictions. And again, like I don't completely, like I'm not used to seeing these kinds of plots, but hopefully if you're in the field, you sort of understand motifs and can interpret them more and what it exactly means. But it's basically, yeah, comparing these the different models and just justifying that yes, what we've done is real, and what we're predicting is doing a good job, better than what's been done before.
If you want a little more depth of what they're doing here, because I personally find this is really interesting. They're taking a stretch of DNA and they're artificially mutating it by substituting sort of some random nucleotides, and then calculating the effect on some of those output tracks as you modify the DNA artificially. So it's like it's almost like artificially CRISPRing pieces of the DNA and making new predictions, and then seeing which parts actually cause a real variation within that megabase on the output tracks. So if you modify a piece of DNA and it changes gene expression a whole lot of a certain gene, then you kind of have some indication that that area actually matters for the expression of that gene, and you can just sort of do that across a whole range of that input sequence, and that's how they're getting variant effect predictions and basically figuring out the causality of variants and things like that. It was really quite interesting to see how they're doing that.
Awesome. Okay, so yeah, they keep justifying. I think they've done a good job at all their justifications in this. So this is I think a similar plot in terms of just but looking at the effect of, wait, what is it, this one? Yeah, what's going on here? So, and predicting the effect of variants on gene expression.
Yeah, so this is where they're testing, basically substituting a nucleotide for another nucleotide, basically causing a SNP, a single nucleotide polymorphism, artificially, and then seeing what effect that has on the output tracks. So they're basically predicting how, are you able to predict what the effect of a mutant is on the expression of genes within that window, and just showing a variety of statistics. And that's a really tough problem. And I forget what the sort of R squared, the correlation, the R value of that was, but I think it was fairly good, and especially the direction of the change of expression. That's what they're showing here in a lot of panels.
There's a lot of panels, yeah. Good. Okay, yeah, they talk about various other effects. I think, yeah, the next plot, this is all about chromatin accessibility this time, and transcription factor binding, so that they're showing, yeah, again, the state-of-the-art performance in causality and effect size prediction for a whole bunch of different QTLs, so they are in strong performance on one of the challenges that they have. So again, yep, just justifying it, I think, and then I think the next one might be, oh, so the next plot, now they're looking at the all the modalities, and they're starting to have a look at the use case that they use for cancer mutations. So that, I think, is this where they introduce the is it the tau1 gene? And yeah, so this was their use case to prove that, yeah, a real life variant, has a, they can measure the effect. Anything else about that?
Yeah, I'll just step in on that one. So yeah, this is a really good sort of example use case of using this in the real world, where they've taken a bunch of known variants around the tau1 gene and predicted basically the effect of some of these known variants. In this case, the yellow one highlighted in panel B is a multi-nucleotide polymorphism, and you can see that it's affecting the expression of tau1. Just off to the left there, you got so just to the left in the expression track, the RNA-seq, the top track is RNA-seq, and that's showing the difference between the reference allele and the alternate allele. And so you can see there's a difference in expression, and then they've sort of shown in some of the other tracks, like in D, you got a heat map there up in D, showing the effect of the variant on the different tracks, and so the different modalities. So the ability to really sort of take a variant, with a known variant, and predict with reasonable accuracy, depending on what you're trying to predict here, to predict what is going to happen due to that variant is quite powerful. And I think, look, there are other models that exist out there from what I understand. I work in plants mostly, not humans, but from what I can see from this paper, this has kind of pushed the accuracy of these predictions to a new high. And so the ability to make in silico predictions of what's happening in the genome, it's pretty impressive. And I'm saying that I'm not a Google person. I don't work for Google. So this full disclosure, but at the moment, this is now the new state of the art on these models.
And so I think the final figure is, oh yeah, so this is talking about everything David was mentioning before, how they did a lot of sort of taking something out of the model and testing it to make sure that making sure that whatever they put into the model, what affected it exactly. So the model design and also the data going into it. And they found that base pair resolution is important for achieving the highest performance. That seems to make sense. And that consisted, one base pair resolution consistently yielded the best results. Training with 1 megabase input sequences and subsequently performing inference using the full 1 megabase context yielded the best results overall. So nothing too unexpected there. And benefits of providing more sequence context during training, and in inference also benefits from longer context. So having that multi-resolution, multi-scale sort of context, I think it is what gives AlphaGenome a lot of power as well. And I don't think the other models sort of had that. They were either sort of high resolution, but a shorter context, or sort of large context with a lower resolution. And also that the distillation using the many teacher models performed well, I think I mentioned earlier. And this is this is basically testing the model in under different conditions and finding sort of optimal parameters. So the resolution, adjusting that and seeing how well the model performs, the sequence length, the ensemble and distillation, and modality ablations, which I don't fully understand what that is, but yeah, but basically they twiddled all the knobs on the model to find the best parameters and to justify that their choices were okay. I think I saw a question in the chat about was it raw data, or did they smooth it all? I think in the methods that they talk at length about every single data set they've used and like what they did for each data set. So it depends on the specific data. So I know some of them they pulled out like just the high quality data. Some of them they used everything as is. So
yeah, there was very extensive quality control and standardization of data. It's I wouldn't call it raw, and it's the methods are enormous. I must say I didn't read it all in too much detail on those methods, but they definitely curate that input data quite strongly.
Any other insights or questions people want to make?
Just seeing a question from Swapan. He says, can AlphaGenome predict trait or function associated with the genetic variant or SNP? It's not trained on any SNP to traits. The only traits it can predict are molecular traits, like the effect of a SNP on gene expression or on chromatin accessibility, methylation states, things like that. It can't predict the effect of a trait on, say, human height or a disease, because it's not trained on that kind of data. But it's up to you as, I guess, the researcher to say, right, this SNP is predicted to affect the chromatin accessibility of this region and the expression of this gene and the binding of that transcription factor there. And then we can go ahead and try and use that combined knowledge to link that through prior knowledge to a certain trait. So I don't know, I don't recall in the paper though if there was any example of actually linking the molecular aspects to a macro sort of phenotype. I don't think that's shown. Pratap has raised his hand. Or is that an accident? We also have Arena has raised hand.
Yes, a quick question. One that I posted in chat on the confidence intervals. So with the AUPRC plots, at least for some of the tasks, I thought it would be doable to have confidence. So let's say figure 4 for the enhancer gene links. I think it's for J. So when you have comparison across models, with Borzoi and RE2G and so on, it's hard to judge if there is an actual improvement over those or not without a confidence interval. For the computational people, do you guys see any reason why that might not be doable? You can always bootstrap the ground truth data and do this 1,000 times and get a confidence interval.
You can open that to the floor.
Yes, I don't know the answer, but in the extended figures, I think they may have taken some of these.
That's still a single AUPRC plot. Anyways, okay, so that's one. The other thing I just wanted to bring up, because it does happen to be my field, the enhancer gene link predictions. I think we do need to be aware of this stage, that at this stage, all those predictions for all of the models are benchmarked on data from a single cell type. So these are K562 cells. So this is a human blood-derived cancer cell line. And all models are benchmarked on enhancer gene links from that cell type. So I think that in the future, having data to benchmark across multiple cell types would be valuable. So it will be very interesting to see how all of these models translate across cell types that they haven't seen. Yeah. So that's just a comment.
Good stuff. Well, I think that will be one of the challenges for the community, I guess, to go and start testing this model out. So we can do that. We can keep asking questions and as we go. But
do we want to keep going because we got a raised hand at the moment, or how would you play this? So, John, do you want to answer that now, Johnny Asaf's question?
Hi, how's it going? Yeah, I guess on the note of the cell types that are being used, it seems nice that the model seems to contextualize what different cell types you can use, like based on the ontologies. I think this will end up being a really good strength of the model later on when it's trained on multiple different cell types. And I think the strength in having an open source kind of model is that the community can also train it on as many cell types as they like later on. But I guess that's kind of different than my question. The question I wanted to ask, and it might be basic, was I think I posted it in the chat. I think I'll just read it from there.
Yeah.
It got kind of buried.
I can see. Do we know anything about how many variants and their interplay can be considered at a time while maintaining optimal performance? Is that the one you're...
Yeah, that's the one I'm referring to. I guess, yeah.
My understanding of it, and I'm happy for anyone else to chime in here, is that you can edit as many nucleotides as you like within a 1 megabase sequence as your input.
Yeah.
And it will model the effect of that. It'll basically, because essentially it's just going to decode your input and produce output tracks based on whatever you give it. And you can, so I think it's no problem making multiple nucleotide modifications. Whether I didn't see any real evaluation of that in the paper. Did any, is that what you're referring to? Does it lose accuracy?
I think it said it increased. Generally, I'm pretty sure I highlighted it somewhere that generally it improved performance the more that you
really
considered. I think so.
But well, when you say considered, like let's say you have your 1 megabase input and you modify, say, 26 different nucleotides in there compared to a control input. So you want to see the effect of
Right.
wild type and then another genotype that has 26 alternate alleles spread out through that megabase. I personally don't recall seeing any analysis like that in the paper, but it's a very long paper.
Yeah, that's why I figured I should probably ask. But I think the heart of that question is just how much variation can the model handle before its predictions start going haywire?
It's a great question.
Yeah.
I want to know the answer to that as well. I don't recall. Does anyone recall seeing any analysis like that in here? I don't.
This is why this is good. How many variants in the interplay can be considered for optimal performance? Let's see if Notebook LM can query.
If you can put it the way I phrased, maybe it might be a bit confusing in terms of phrasing.
Yeah, the phrasing here kind of matters.
Yeah. I can see it.
I think what you're referring then, Johnny, is that the more variants you do adjust at once, things like epistatic effects and play, just linkage disequilibrium effects and all of these things start to come into play, and has it been trained enough on the ability to get joint effects like that going? I don't know.
Okay.
Sorry, so it's designed to analyze individual genetic variants. So maybe that's
Yeah, but there is nothing to stop you as the user from modifying multiple individual variants in a 1 megabase sequence. It's just, is that a bad idea at the moment with this model? I think that's actually a really good open question that I wonder if we can get an answer to that later from the Google boffins.
So one of the links I also posted was to a YouTube video that was all from the team at DeepMind, giving a great sort of introduction to it and everything. So I think some of these questions might be incident.
Is this the 101? The one on
AlphaFold 101.
That was good. Yeah.
Okay, so just in the interest of time, I might just push to some sort of demos. So in there's a whole bunch of comprehensive documentation. So AlphaGenome is kind of like two parts. It's the model that does kind of like all the really hard work, but then there's this code and a software development kit around it, or a Python package that kind of wraps it and allows you to query that model sort of easily. And so this documentation is all about the code and how to interact with that model. So yeah, there's kind of like two parts to it, and there's a few. I'll just post a link to it in the chat. There's a really good quick start notebook here, which steps through kind of some fundamentals in the Python code required to sort of interact with the model. I won't go through this one because it's covered in that video that I just mentioned, and it's also really nicely written and stepped through. So you can do that in your own time because I think it's quite rewarding. Instead, I'll go through I think what I found to be more of maybe a real analysis where it's a bit more hacky and a bit more thrown together. But throughout this, there's a bunch of tutorials for when you actually need to do your own work. Hopefully, one of the tutorials here or one of the user guides will steer you in the right direction. These are all different Python notebooks and workflow analyses that you can copy. And the FAQ is also quite comprehensive. Now, I was going to just demo this example analysis workflow, Tau1 locus. And I'll show you sort of briefly kind of how to do that. If you've, if you have no Python experience, it's okay, because you can just kind of click next, 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 and it will, and hopefully you can just sort of sub in like your pieces into it in the future. So this is called the Google Colab environment, and it's basically a way to run Python in your browser. And the first thing you're actually going to need, let me just click on things. So we're going to need an API key, and so that gives us access to the model. It's basically like a password to the model system. So you may have used API keys to get access to sort of anything where you write code to query some sort of server where they keep data. Instead of using a username and password, you can use an API key. And to do that, you go to this website and say, please give me an API key, and you click create API key, and it should make you one. This one will say it fails because it doesn't give me an account, because I work at Google, which is funny enough. So I have to use a personal account. Now, once you get an API key, we'll plug that in a minute. I'll show you how to do that. And let me see what else is important before we sort of kick off. Oh, the other thing you might want to do is go File, or you can click this button and click Copy to Drive, and that will make your copy sort of personal. Otherwise, once you close it, you'll lose sort of all the information as you're using the DeepMind one. So if you make a copy of it, then any edits you make will become permanent. Okay, so yeah, so in this, we'll explore non-coding T-cell acute lymphoblastic leukemia associated with mutations near the tau1 locus. So we'll look at the variants around that area and see like what, I don't want to use the wrong language here, but what turns on the cancer. So yeah, the T-cell acute lymphoblastic leukemia is a cancer of blood and bone marrow. A large fraction of all cases arise due to aberrant upregulation of the tau1 oncogene. In particular, locus harbors positions in which non-coding variants can occur that activate tau1 gene expressions from several kilobases away. So that's that's what we've been talking about, what it does, and this is the example of how to do it. So we just do some visualization, and then we'll do some predictions as well, and then we'll compare the predicted effects of known disease-associated variants with a set of background variants. All right, so step one in this is to install AlphaGenome. You can do this also on your local Python. That piece of code there, the pip install AlphaGenome, installs AlphaGenome. That installs it from a remote repository, which is just a public repository. That's how you install other packages in Python. And so once it's installed, it should take a minute. So it installs this AlphaGenome package that DeepMind has written along with the code. So it's successfully installed it, fantastic, and it hides it once it's installs it. I'm going to run the second cell here, so I can click play, and this loads in all these different components from that Python package, and then a bunch of general Python libraries as well. So if you're familiar with Python, then that should be straightforward. If not, you can just click the play button and get a tick there, and it should be good to go. Okay, now this is where we need to load in our API key. So this thing, DNA client.create, is in here. Let me search DNA. I'm in the documentation. So this basically just, you put your API key in there and load it in. So it will look something like, A, B, C, blah, blah, blah. It looks something like that you should hopefully get from the DeepMind page, and you copy it and paste that in there as a string, and then you should be good to go. Now mine is saved, grant access. So mine's just saved in like a secret file over here, so I can read it directly using that little bit of code. But if you don't have that, you can just paste it in. But if you're just using it for yourself, that should be secure enough. Okay, so that's probably the hardest part, I think, technically. So once you've got your API key and plugged it in, you should be ready to get going. Okay, now we get to some fun things. We're loading in gene and annotations from GenCode. So that's just a public repository, and we're loading it. We want to filter out a whole bunch of some details that we don't need. So this loads in something. First, it loads in this thing called GTF, which is a huge data frame. I think it's yeah, what is that? 3 million rows or something. So that's too big. So we just filter it down until it's something manageable, because we just want we want the highest quality data here. So I think they yeah, that they have some flag on there that says just give us highly supported transcripts, and then also we want the longest transcripts of those high quality ones. And so we're slowly filtering out the data. We've got 438,000 rows now, and we'll use that as a as sort of a baseline as we move forward. Okay, so yeah, that's not too important. We'll come to that later. Now, I feel like this is really probably real. It's somewhat, I think, confusing code, but you set up these like functions that we're going to use later, and what the most important thing that we do is we want to later on we're going to have our variant which causes the cancer. Again, I'm careful of my language, or not careful of my language. And we want to compare that to a whole bunch of just random variants. So we make this function that basically just creates random variants for us. So I guess there's no built-in function into the AlphaGenome package. So we just write some code that sort of does that on our behalf. And I think that this whole cell basically does that, and converts it into the right formats and everything. So it's quite large, but that's the important part for that. Okay, and then we load in some data. So this, you'd probably actually read this from like a CSV file in real life, but they've just sort of written it all down here. But it's the TOR variants that affect the tau1 gene. So that's the data frame, and it just looks a bit like this. So that's the data that we're reading in. But yeah, in real life, you'd probably read that in from a CSV file or something. Okay, and then then we get to some fun parts. This is where we actually start to visualize it. So I'll just run this cell, and you can see the plot of it. I think so. Yeah, from the plot we can see the tau1 gene is transcribed from right to left as it's on the first strand, and we've got 3 different clusters here. So there's a cluster of 5 variants upstream of tau1. Then we've got an intronic variant and a 3 cluster of 2 variants downstream. Okay.
So as far as I can tell, that plotting is part of the API, right?
That's right. Yeah. So it has these built-in plotting functions, which again are pretty confusing, but it is documented. So if you love Python, you should be okay. If you're new to Python, this is actually confusing, but it is documented of like what each of these things does and how to use it. But in the first instance, you can just sort of copy and paste what's going on here. I think besides the plot here, the other interesting thing which sort of drives the whole kind of package is this line here. So what this does is it defines an interval. So so you just set a starting point and an ending point. And I think that number minus that number is about 32,000. So it's 32 kilobases long. So, and yeah, we're on chromosome 1 strand, the first strand, and yeah, you define that interval, and then everything is sort of built around these the sequences that look like that. Okay, so we plot it, look at it, fantastic, and now we start to plot the predicted effect of these variants that we can see here. Okay, so let's do that. We defined an ontology term here. So that's the ontology of interest. I'm not fully clear on what that is, but hopefully people know what that means. And yeah, okay, so to make predictions relevant to the specific biological context, we provide the model with an ontology term and and so it comes from there, and it's a close match to the tissue of origin reported there, and which use purified hematopoietic stem cells on a microexpression. Okay, and so to make predictions, again, we'll specify another interval. So that's that interval data type that is sort of inherent to the AlphaGenome Python package, and we make it 1 million base pairs long, which is about 2 to the 20. So we'll also pass in our the requested output. So what kind of modalities do we want out on the end? So we set this thing up here for what we want to predict. So we set an interval, how long it is, the variant, which we defined earlier, I think. No, we haven't done that yet. Oops, sorry. And yeah, those output types. Okay, so run that. And now I did that. Okay, and so that, I think, connectors to the API. So that's where we actually went to the API, made a request to the model, sending all these different options there. So we sent our interval, we sent the variant of the interval, and we sent the outputs that we want. And if you just look at the output, it's probably just yeah, it's a dump of stuff, but we can access it in a few different ways. Okay, how are we doing for time? So again, this is building plot, it's all just plotting, which is confusing, but that's what it does. And so we get out this beautiful plot. So that's what AlphaGenome does. I guess there aren't probably not too many Python packages, definitely not by default, that let you sort of make these plots nicely. So it's pretty cool that they've developed this as well. And so this shows the difference between the model predictions for the alternate sequence in which the alternate base looks like this, where they've been inserted into the genome at chromosome 1 position at that, oops, sorry. So here basically, and so yeah, interpreting the plot, these are the outputs as well that we requested. So that's what's sort of shown on that plot. Okay, and then finally we can compare the predicted tau1 expression change for cancer-associated variants compared to randomly shuffled variants. And so that was just making the prediction, but now we want to really test it with our what happens if the variant was just random instead of that specific one, which is we already know the answer for. And yeah, again, it's just Python code setting ourselves up with a random shuffle of stuff, and we can now send instead of doing one prediction, we can send a whole bunch of predictions all at once to the DNA model API. So here's where we call the API. I'm just going to do that. I'll run that. And yeah, so this is pinging the API a bunch of times. And hopefully we get an answer. All right, so then I think the rest is also just plotting as well. All right, so maybe I'll wrap up there, but by all means, go through that first, the quick start notebook, which is really nice and has nice diagnostic plots as you go. But hopefully this is sort of enough to get you started. If you want to use it, you've got an API key, you got access to the code now, so you can start subbing in your own kind of your own data. Any final questions before we kind of wrap up whilst my code is pinging? All good.
There's been a little bit of debate in the chat about the ability to do cell-specific predictions, cell type specific. I'm not 100% sure where we lie with that. I know there was, like with GTEx and things, there's tissue-specific gene expression. It was trained on tissue-specific gene expression, and you can predict different tissues, and that, I think that's why you put in that ontology term to give it context about where you're trying to predict. I'm not sure how in-depth that is at the sort of cell type specific stuff. Has anyone got any input on that?
Yeah, I think it's safe to say that there isn't any imputation of the unobserved parts of the ENCODE tissue by experiment matrix.
Okay, so that's kind of like a no in terms of.
So you can predict on any tissue type that it's seen, any experiments that it has seen on that tissue type.
Right.
I think it's kind of like asking, can it predict something that we haven't tested if it can predict or not? And like, it's just, sure, you can try, but we don't know if it works, right?
Yeah, you'd need some level of evaluation beyond what's done in the paper here to be confident of that actually working.
I look forward to people trying it out and seeing papers published, which would be cool. Okay, so in the interest of time, we'll wrap it up there. But yeah, thanks everyone for coming. I've definitely learned a lot through this experience. We have a bunch of other events coming up. Maybe not bio related, but fascinating nonetheless. And we'll probably do a few more of these journal club styles. So we're doing some Python workshops, some R workshops. We've got a couple of climate workshops coming up, data set workshops, and some indigenous large language model stuff as well. So lots happening in the community. So thanks very much, everyone. And yeah, the recording will go up on the GDG page and also on our YouTube. All right. See you all soon.